Good evening, guys. Today, we'll be starting with the week 7 of RNA biology lectures. I hope I am audible and the screen is also visible. I hope my screen is visible.
Yes.
Today, we'll be starting with a new topic, that is eukaryotic transcription. In the previous lectures, we have completed about the prokaryotic transcription and how it is regulated uh, in the form of operons. So we have studied lac operon, tryptophan operon, and some a little information about the bacteriophage lambda, the transition between the lytic and lysogenic cycles. So this we have covered in the previous lecture. So just a recap. So talking about the structure of the eukaryotic gene. So the basic structure of the gene is the starting with the promoter. We have a RNA polymerase binding site. This is basically a promoter where the RNA polymerase binds, recognizes a region on the DNA, and then we have the structural gene that is being transcribed into RNA. And then we have a terminator where the RNA polymerase basically separates out from the DNA and the RNA is released into the nucleus for further processing. And talking about the eukaryotic gene structure, so basically in prokaryotes, as we studied that this is a promoter that is linked to various genes. So this will be gene 1, this will be 2, followed by many, and then will we have the terminator. So basically, when we have a similar kind of genes that perform a similar function or function in a particular pathway. For example, in lactoperon, we have genes that help in metabolism of lactose, and in tryptophan we have genes that produce enzymes that are linked to the production of the tryptophan. So similarly, all kinds of genes followed in a similar pathway are linked together to form an operon. But in case of a eukaryotic gene structure, what happens? One gene is only linked in a particular promoter. So we form only one protein from one particular gene. But in case of prokaryotes, we have multiple proteins from one single mRNA. So that is why the mRNA formed from prokaryotes is called as polycistronic, because it is formed of multiple genes. And then in case of eukaryotes, we have only single gene linked to a promoter, so it's called as a monocistronic mRNA. And the major thing in eukaryotes is a gene has various intriguing sequences. These are called as introns, and the exons are the major coding sequences. So once the mRNA is formed, the intron sequences are removed from the mRNA, forming a mature mRNA that contains the continuous sequence of the exons. These introns are the non-coding sequences. They have majorly role during the regulatory processes of the expression, not the protein, not the coding part. Then for starting the transcription, we have the promoter. So promoter basically has two kinds of regions. One is the core promoter, where the basic RNA polymerase binds. So what happens, as we have previously discussed, there are two kinds of promoters. One is the weak promoter and other is a strong promoter. So strong promoter is generally present in prokaryotes, where it does not need any activator or accessory proteins to help in the transcription. Okay, the general funda is the strong promoter sequence is nearly same to the consensus sequence of the organism, and in case of the weak promoter, the sequence is very different from the consensus sequence, and it is generally present in eukaryotes. Okay, and here what we have certain extra sequences also that are linked to the activators. So why eukaryotes have these weak sequences and why prokaryotes have majorly strong promoters? This is has some relation with the complexity of the organism. Okay, then due to which the eukaryotic promoters are weak. Do you have any questions?
12,
Sorry.
12, 12,
Uh, Okay, so the activators are generally not required in strong promoters because the RNA polymerase can alone bind to the strong promoter, and it can actively transcribe the RNA. But in case of weak promoters, what happens? The RNA polymerase cannot bind to the promoter in that intensity. So what happens? The frequency of formation of transcripts is really less. So that is why we require a activator kind of molecule that can basically intensify the transcription process if needed. So this proximal control elements are the major binding site for the RNA polymerase and all the transcription factors that are involved in helping the RNA polymerase to bind to the promoter. And these distal control elements are the binding site for the enhancers or silencers. So silencers basically helps in the regulation of the transcription. So if they are bound to the particular promoter, so they will not allow the RNA polymerase to complete the transcription. So these are basically helps in regulation. And enhancers, as I talked about, these are activator sequences that help to intensify the transcription process. And then to complete the transcription, we have the terminator sequences. So the terminator is basically the site where the RNA polymerase detaches from the DNA, leaving behind the RNA for further processing in eukaryotes. So here, in case of eukaryotes, it is linked to the addition of the poly A sequence that we'll study in further slides. So this was the basic structure of the eukaryotic gene. And moving on to a bit in detail about the promoter sequences. So as we discussed, we have a weak promoter in eukaryotes, and the promoter depends upon the RNA polymerase. So here, generally we have three kinds of RNA polymerases in eukaryotes. One 2, and 3. These are the namings for the RNA polymerase. We have 4 and 5 also, but that are not studied intensively yet. But these three are major, and we have three different promoters for these three different RNA polymerases. And these, in case of eukaryotes, there is a feature that first the transcription factor binds to the promoter, and then it recruits the RNA polymerase. Okay. So this is the main feature of the prokaryotes, and in case of in eukaryotes, and in prokaryotes, what happens? We have a sigma subunit of RNA polymerase that helps in the identification of the site and recruiting of the whole RNA polymerase. So this is the basic difference between the eukaryotic and prokaryotic transcription initiation. And followed by this, in case of prokaryotes, as we have learned that this is the transcription start site. So this is considered as plus one, and in before that this is the upstream region, and after this is the downstream region. So in case of prokaryotes, the minus 10 box and the minus 35 box was present upstream of the transcription start site only, and not downstream. But in case of eukaryotes, it is present both upstream as well as downstream. And it depends mostly upon the RNA polymerase type, that what kind of promoter is present. Okay, that we'll see in a bit detail. So starting with the RNA polymerase 1 promoter. So it basically has two regions where the basic transcription site starts. So one is the core promoter and other is the upstream control element, that is UCE. So core promoter is like in between the transcription start site. So it is like the transcription start sites basically starts from the core promoter. Then it is located between the minus 40 and plus 20 region, and this region is basically AT rich in nature. Why? Because from here only the transcription bubble starts. And as we have discussed in prokaryotic transcription also, where is the transcription bubble would be present or starts, that region would be mostly AT rich, because it has only two hydrogen bonds. So it would be easier to break these bonds and form a transcription bubble. Okay. Then we have the upstream control element. This is the GC rich region, and just one thing you can remember, that mostly the protein binding sites which are activators or regulatory elements that are not involved in the transcription bubble formation, those regions are basically GC rich. Okay, and these regions are the binding site for the activators or enhancers. So, if these regions are mutated or basically removed from the RNA promoter, so what will happen? This will not hamper the transcription, but it will hamper the transcription rate. So, if these sequences are not present, the rate of the transcription would decrease, and these help in increasing the transcription rate. Okay, and the region between which these are present is minus 100 and minus 80, so 180. So this is upstream of the TSS. Okay, so any doubts still here? No, RNA polymerase doesn't bind to this UCE, it basically binds to the core promoter only. So basically, we'll study about the RNA polymerase 2 and 3 promoter. It basically depends upon the what kind of RNA they are producing. So, for example, RNA polymerase 2 generally produces the mRNA. So these regions that are decided as the promoter sequences are based upon the product size of the all the RNA polymerases. For polymerase 2 and polymerase 3, we'll study the promoter in detail in the next few slides. Okay, if there are no further questions, we'll move on to the RNA polymerase 2 promoter. So, this is the most extensively studied promoter, and it has basically four regions. One is the part of the core promoter, two regions are part of the core promoter, and two regions are part of the regulatory sequences. So, talking about the core promoter, so in each of the promoters, we have a core promoter and a regulatory sequence. The core promoter is the region where the RNA polymerase binds and start the transcription. So here we have one INR sequence that is overlapping with the transcription start site. So some of this region is upstream and some of this region is downstream. So it extends from -2 to +5, and it has a sequence of this is a consensus sequence. It has two pyrimidines, a at the +1 site, that is the transcription start site, and then we have any of the nucleotides followed by two pyrimidines. So this was generally found in all of the RNA polymerase 2 mRNAs. So that is why this is the uh, consensus sequence form. Then we have a Tata box. So this is present at the -30 region, or this is also called as a Hockness box. And this is the main region from which the RNA polymerase binds. Okay, the first transcription factor is recruited to this particular Tata box, and further the RNA polymerase is recruited to a bit of the transcription factors. The full process we'll see in detail. And a part of the core promoter, we have the regulatory sequence. So two regulatory sequence have been studied till now. One is the GC box that have a consensus sequence of triple G C double G. This is the very strong sequence that is present in the eukaryotic RNA polymerase 2 promoter, and it is present at the -90 position. And then we have the CAAT cat sequence that is present at the -70 region. And these three sequences work in orientation independent manner, means that these sequences might be present like interchangeably, that cat sequence is present in place of GC sequence or vice versa, or either of the sequence can be present and either of the sequence can be absent. So in the both ways it can work. Okay, these are the part of the regulatory sequence. So this was the major thing about the RNA polymerase 2 promoter. Then moving on to the RNA polymerase 3 promoter. So this is the RNA polymerase promoter that is least steady. So it depends upon the type of the transcript it is forming. So RNA polymerase 3 forms three types of transcript. One is the U6 snRNA, 5s rRNA, and tRNA. So based upon the transcript, its position changes. Okay, so RNA polymerase 3 promoter is divided into two types. First is the upstream located and the other is the downstream located. So this is based upon the transcription start site. So for example, here is the transcription start site. This is the upstream and this is the downstream part. So for this particular thing, it's the upstream located promoter, and for this thing, it's the downstream located promoter. So the upstream one is called as the type 3 RNA polymerase 3 promoter, and it produces U6 snRNA that is basically linked to the spliceosome machine. And the downstream located promoters are present after the TSS, and these are type 1 and type 2. So type 1 produces 5s rRNA, and the type 1 produces 5s rRNA, and type 2 produces tRNA. So there is a difference in the sequences and the number of boxes present in either of the promoters For example, in the type 3 promoter, we have two sequences apart from the Tata box at minus 30 position. We have OCT, that is octameric sequence, and the PSC, that is proximal sequence elements. So these are upstream of the Tata box, and these are regulatory sequences, that is binding site for both activators and silencers. Followed by the type 1 and type 2 box, we have box A and box C uh, after the transcription start site, and we have box A and box B in the type 2 RNA, type 2 RNA3 polymerase. Okay, these both are downstream of the transcription start site. But like the major information about these promoters is only limited to these particular sequences, not further information is available, and research is going on about these promoters. So any problem till now about the RNA polymerase 2 and 3 promoters? So if not, then we'll move on to the next topic, that is nuclear RNA polymerase. So a description about the RNA polymerase in the eukaryotes. So starting from the like we have two kinds of RNA polymerase present in the eukaryotic system. So just a brief. This is a eukaryotic cell. We have a well-defined nucleus. Then we have two kinds of organelle that are basically involved in their own transcription due to the endosymbiont theory. So this is the mitochondria, and the other one is the chloroplast. Okay, so due to which we have two kinds of RNA polymerase. One will be located in the nucleus. So we have the nuclear RNA polymerase, and the other two are present in the organelles. So we have organelle RNA polymerase. So we'll be studying about the nuclear RNA polymerase first. So in this case, we have five kinds now. We have four and five also. These are mostly located in plants. Sorry. So these are mostly located in plants. So starting with the first RNA polymerase, it produces the rRNAs. Okay. So it produces 28S, 18S, and 5.8S RNA from a 45S rRNA precursor. So it has a large rRNA precursor from which these three rRNAs are basically excised out. Then we have the RNA polymerase 2, which produces mRNAs. So this is the primary mRNA and this is the mature mRNA. Then RNA polymerase 2 also produces the snRNAs, small nuclear RNAs. Okay. Then we have RNA polymerase 3 that produces 5S rRNA, tRNAs, U6 snRNA, and these two regulatory RNAs. Okay. So to remember, all the rRNAs are produced by the RNA polymerase 1, the mRNA is produced by RNA polymerase 2, and tRNA is produced by RNA polymerase 3. This is the general question that is asked in the competitive examination. Okay. And then moving on to the 4 and 5. So 4 RNA polymerase is related to the siRNA production in plants, and 5 is related to the regulation of this siRNA. So these two RNA polymerases are discovered in plants yet, no signs in animals. Then there is a distinguish between all the three RNA polymerase. So what happened? There is a mold called as amnesia phalloides. So this produces a toxin called as alpha amnatin. So all the three RNA polymerases are a bit or like are differentiated on the basis of the sensitivity towards this toxin. So based on this figure, we can figure out that RNA polymerase 1 is least sensitive or basically insensitive to alpha amnatin. Okay, and then RNA polymerase 2 is the most sensitive to alpha amnatin and followed by the RNA polymerase 3. So RNA polymerase 2 is 1000 times more sensitive to alpha amnatin as compared to RNA polymerase 3. This was the sensitivity towards the different RNA polymerases of due to alpha amnatin. Okay, so this is a basic thing to distinguish different RNA polymerases on the basis of toxicity. Uh, Sorry, I'll check for the question. Does mitochondrial DNA follow transcription style as in prokaryotes, or is it identical to eukaryotic nuclear transcription machinery? So, yes, it follows the prokaryotic transcription style. We'll study in a few slides about the RNA polymerase. From that, you'll get an idea about the how it follows the transcription. So, it basically follows the prokaryotic style. So, is this point clear about the sensitivity of the RNA polymerases towards alpha amnatin? So, moving on. So, the basic theme of the like universes, we have all the like proteins that are present both in eukaryotes and prokaryotes. These are particularly linked. Okay, the functions is similar and all kind of stuff. So likewise, this is the structure of the RNA polymerase present in the prokaryotes. Okay, so we have only single RNA polymerase present in prokaryotes, but different kind of RNA polymerase is present in eukaryotes. So this is the prokaryotic RNA polymerase. We have beta prime beta subunit involved in uh, yes, they have a specific mechanism from which these are immune to their own toxins, but the detail I am not clear about yet, so I can check and let you know. So in the prokaryotic RNA polymerases, we have beta prime and beta subunits involved in the polymerization, alpha 1 and alpha subunit that are basically involved in the um, stabilization of the subunits as well as binding to the upstream element. And then we have the omega subunit. Omega also help in the stabilization of the subunit after binding to the after forming the core polymerase. So similarly, for RNA polymerase 1, 2, and 3, as the complexity increased in the eukaryotic system, so we have different kinds of polymerases. So talking about the different subunits, so beta and beta prime like subunits, we have two subunits in each of the polymerases that are similar to the polymerase subunit in the prokaryotes. Similarly to alpha and omega like subunits. So, talking about the different number of subunits present in 1, 2, and 3. So, in 1, we have 14 subunits. In 2, we have 12 subunits. And in 3, we have 17 subunits. So, these are the number of subunits present in each of the RNA polymerase, some of which are only applicable to that particular RNA polymerase, but that might not have any role in the RNA synthesis or binding to the promoter. They might be regulatory subunits, like additional enzyme specific subunits mentioned here. Okay, so for simplicity, we mention them only the numbers and the type of subunits that might be present. Okay, so moving on to the main RNA polymerase, that is RNA polymerase 2, because it is well studied and there is a peculiar structure present in this particular RNA polymerase that makes it a bit interesting to know. So this is the structure. It has 12 subunits in total, and there is one polymerase subunit that have a extended C-terminal domain that is called as a tail. Okay, as you can see in this particular structure, this is the C-terminal tail of RNA polymerase 2. Okay, and it has a repeat of heptapeptide sequence that is tyrosine, serine, proline, threonine, serine, proline, serine. So the serines and threonine are basically phosphorylation sites that are being phosphorylated by different kinases, and the TF2H is the major kinase that have been found to involved in phosphorylating these particular serine residues. So what happens whenever a different step needs to be followed? For example, while forming of the pre-initiation complex, when it has to be transferred to initiation, then different number of serine residues are phosphorylated such that it can start the transcription, and it can recruit different transcription factors that will help in transcription. And secondly, as the RNA processing starts, so RNA processing is co-transcription in eukaryotes. So it these phosphorylation sites at different serine residues will recruit the different enzymes for capping, splicing, and tailing also. So this phosphor tail is important in recruiting different proteins. Okay, so that can be involved in regulation, that can be involved in RNA processing. So this signaling also is important in the point of view this carboxyl terminal tail. No, the subunits are involved only in the RNA polymerase 2. These are different proteins that are involved in different processes during the transcription. These are not subunits that are being recruited. And then these number of repeats, so this was the heptapeptide repeat. So different organism based upon the complexity will have different number of repeats. So for example, humans have 52 heptapeptide repeats in their carboxy terminal tail, and there are different number of repeats in drosophila, different in plants, so on and so forth. So based upon the complexity and the transcription style, these have different number of heptapeptide repeats. So this is the enzyme that have been extensively studied, and this is the peculiar feature, the carboxy terminal tail, that is like a fascinating feature of the RNA polymerase 2. Now moving on to the organelle RNA polymerases. So there are two kinds of organelle, mitochondria and chloroplast. So in both the organelle, we have different subunits that are being encoded by the RNA polymerase, genome of that particular organelle or the nuclear genome. For example, in mitochondria, the R, sorry, the RNA polymerase is totally the subunits is encoded by the nucleus, and the organelle RNA polymerase is nearly similar to the prokaryotic RNA polymerase. Okay, so it will have all the sigma, beta, beta prime, alpha 1, alpha 2, and omega subunits in it. Okay, and these subunits, except the sigma subunit, is called the core subunit. So both core enzyme and the sigma factor are derived from the nuclear genome. So total full RNA polymerase is encoded by the nuclear genome in mitochondria. And in chloroplast, we have both the subunits like chloroplast as well as nuclear encoded. So the core component and the sigma factor both from the nuclear genome. So in some of the chloroplast, we have core component encoded by the chloroplast genome, and the sigma factor is encoded by the nuclear genome. And in some of the chloroplast, we have full components, that is nuclear genome encoded. Okay, and both the RNA polymerases are similar to the bacterial RNA polymerase. So I have one question to all of you. Just check on the fact that this RNA polymerase is sensitive to alpha amnatin or not. So you guys check and let me know tomorrow. Now this uh, ends with the part of the RNA polymerase. Now moving on to the different transcription factors that play a role in the RNA polymerase initiation. So starting with the transcription factors are DNA binding protein. So how can we detect the transcription factors that are being bound to the DNA? We have different processes, like we have uh, EMSA that we have studied, DNase binding assay or DNase footprinting. Then we have DNS footprinting. So all these we have discussed in the previous lectures, and how these will help to detect the DNA binding to the protein, to the particular protein binding to the DNA. So starting with these particular proteins that we see are transacting in nature. So what is the meaning of transacting? So this means that if this protein is present in vitro, for example, and this particular DNA sequence that it recognizes is also present, then this protein, irrespective of the environment, this will bind to this particular sequence. That means wherever this protein finds this particular DNA sequence, it will bind. And being this DNA, eukaryotic DNA or prokaryotic DNA or any of the organism that is present, so if this protein finds the sequence of like binding, that will bind to that particular sequence irrespective of the environment, irrespective of the source. So this means that it is transacting in nature. Cis acting means that it will bind only to specific proteins. Okay, for example, there are some proteins that only bind to the sequence that are present in only one organism or the same organism, rather than binding it to from different organisms. So those proteins are basically cis acting in nature. So generally, the sequences that we have, the DNA sequences, are cis acting in nature, and the proteins are transacting in nature. Okay, so moving on to the transcription factors. So there are two kinds of, yeah. So cis acting means that it will bind to it is for example, it is organism specific. So there are some proteins, like they will only bind to sequences that are present in same organism. Okay, so this means that those proteins are cis acting in nature. And like we can see that DNA sequences are basically like specific to specific organisms. Okay, so that means the DNA sequences also are cis-acting in nature rather than proteins. So proteins are trans-acting in nature because they can bind to the same sequence, whether it's from the same organism or from a different organism. So am I clear? Yeah, it might not bind to that particular sequence if it is present in from another organism. Okay, so shall we move forward? So we have two kinds of transcription factors. One is the basal transcription factors and the regulatory transcription factors. So basal means that will help in initiating the process of transcription directly. So for example, this is the promoter. First, the transcription factors bind that will recruit the RNA polymerases. So these transcription factors are the basal transcription factors. Okay. For example, we have TF2D, TF2A. So these are all the transcription factors which we will be studying for RNA polymerase 2. So they form the basal transcription apparatus. Okay. After recruiting the RNA polymerase. Then we have the regulatory transcription factors that regulate the process of transcription. So we talked about the activators, enhancers, and silencers basically. So we have two types, the activators and the repressors. Activators help in increasing the transcription rate of weak promoters that we studied, and repressor basically responds negatively. They negatively regulate the transcription rate, and these are basically involved in prokaryotic promoters because they are the strong promoters. Okay. So some of the examples for from regulatory TF transcription factors, we have the CAT binding factor. So CAT is present in uh, RNA polymerase 2 promoter. Okay, so this CBF binds to CAT, and this also regulates the process of transcription. Then we have CAT or enhancer binding protein that enhances the process of transcription in RNA polymerase 2 transcription. Then we have cyclic AMP response element binding protein. So this basically binds when cyclic AMP like is high or low, as we have seen in the lacoperon. Okay. Then we have SP1 bind protein that bind to the GC box. So these proteins are basically involved in the RNA polymerase 2 transcription. So there are many regulatory transcription factors that are involved, but we have, for simplicity, we have named a few so that that will be easier just as for you to understand as an example. Okay. Then now till here there is any doubt, so we can move on to the next process, the process of eukaryotic transcription. If you have any doubt, you can ask.
12, 12,
Um, Sorry, I'm not able to understand. You can type in the chat box.
12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12, 12,
Sorry. So, if there are no questions, we will move forward to the process of transcription in eukaryotes. So, first, we see about the pre-initiation complex. Okay, so there is no ratio kind of thing in the transcription machinery, but they might be present in equal ratio as far as my understanding is there, because the basal in my opinion, the basal transcription factors would be more in number as compared to the regulatory transcription factors, because there are a lot of genes involved here, and the number of basal transcription factors are more as compared to the activators. So, like, in my understanding, this would be the case, but I'll let you know about the actual concentration if there is a number present or not. Hmm. Sure. So, starting with, so first, what happens in the eukaryotic transcription? First, there is a pre-initiation complex formation, followed by initiation, then followed by the normal process of elongation and termination. Okay, so starting with the pre-initiation complex, what happens? First, the transcription factor binds. So the first transcription factor that binds to the DNA is TF2D. Now, how these are named? So TF means transcription factor, 2 means it belongs to the RNA polymerase 2 transcription factor pool, and D is basically the naming based upon the how they are isolated and how they are discovered. Okay, so this is the naming. So the first transcription factor is TF2D. Now TF2D has two parts. One is the TBP and others are the associated proteins. Okay, so TBP stands for Tata binding protein. So this is a subunit of the TF2D only. So it is a large transcription factor that have two domains. First is the Tata binding protein that recognizes the Tata box. Okay. Then apart from the TBP, it has several associated proteins that helps in binding as well as stabilizing the TF2D structure. So this was the first transcription factor that binds to the DNA. Then comes the TF2A and TF2B. So these TF2D is further bound by TF2A and TF2B. Now this particular complex recruits the TF2F that is bound to the RNA polymerase 2. Okay, so this TF2F helps in recruitment of RNA polymerase 2. This TF2F has a binding towards TF2D. That is why it helps in recruitment of the RNA polymerase 2. Then comes the TF2E and TF2H. So TF2H has a kinase activity. So this basically helps in like extracting energy from ATP. Okay, and TF2E has a role in like opening this DNA chain. Okay, so now when all of these subunits are basically recruited, then the RNA polymerase starts the transcription. So this RNA polymerase, why these transcription factors are needed? Because this RNA polymerase do not have a binding site on these particular promoters. Okay, so these transcription factors helps in properly recruiting the RNA polymerase, and then it will like stabilize the DNA and the protein complexes further, so that it will start the initiation process. Okay, so this was the case. So first TF2D comes, then TF2A, then B, followed by F and the RNA polymerase 2, followed by E and H. Okay, so this was the sequence in which the RNA polymerase are bound. Sorry, the transcription factors are bound. So please remember the sequence, because many a times these particular questions are asked in the competitive examinations directly in the form of fill in the blanks or any of the questions they can ask. Now what happens after binding of this RNA polymerase? This final complex is called as the pre-initiation complex. Now TF2H plays its role, and it phosphorylates the specific serine residues in the C-terminal domain of the RNA polymerase 2. Okay, this activates the enzyme, and this basically starts of the initiation of the transcription. So initiation starts from the INR sequence present at the transcription start site. Now, as this binding of transcription factor to the promoter is very strong in nature, okay, so it is a really difficult task for RNA polymerase to leave these transcription factors and move forward for elongation. So what happens? There is a process called abortive initiation. So what happens during initiation? There are around 10 to 12 nucleotides formed, and then the RNA polymerase slips off from the DNA. This leads to the stoppage of the initiation, and then the elongation also stops. Further, the new RNA polymerase all again is bound to the DNA, and the process of the initiation starts. So this happens till the RNA polymerase continues the RNA length in during the process of elongation. Okay. So this is the process of abortive initiation that is also seen in prokaryotes also, till the RNA polymerase leaves the promoter in a proper way. So this is the process where the transcription factors are like leave the RNA polymerase, so that it will continue the process of elongation. So this is abortive initiation is the process where the RNA transcripts, short RNA transcripts are left, because the DNA polymerase cannot stably fit on the DNA and complete the process of initiation. So this is because the binding of the RNA polymerase with the transcription factor is so strong, so that RNA polymerase cannot leave those transcription factors. So once the RNA polymerase leaves these transcription factors, this leads to proper and successful initiation process followed by the elongation. So this was the abortive initiation. Okay. So any doubt in the initiation and the pre-initiation complex? Yeah, you can ask. would it be possible for you to briefly mention or give references as to how experiments were designed to figure out the sequence of binding of transcription factor in the pre-initiating stage of transcription? Okay, it is not possible now, but yeah, I can let you know. Yeah, so that also happens. So once the abortive initiation, like once the RNA polymerase falls off from the DNA. So for example, this was the DNA, this was the RNA polymerase. So first it transcribed only 10 to 12 nucleotides and then it falls off. Then again it is recruited to the transcription factor pool. This is not dissociated during abortive initiation. Okay, so because they have the signal to bind to the promoter. So now only the RNA polymerase is recruited and then it again starts the initiation. Am I clear with the answer? No. The thing is, this binding is so strong and they have the particular signal to bind to the promoter. So they don't fall off during abortive initiation. So what only the RNA polymerase is aborted. That's why it only now like bind to the transcription factor that would again start the process of initiation. Yeah, it would be binding. The transcription factor would be binding because only the transcription factor has the affinity toward the other transcription factor as compared to the RNA polymerase. So transcription factor will be bound to the RNA polymerase for its recruitment. So any more doubts? So this happens not only one or two times, this happens many times. Okay. And what happens once this abortive initiation is over? So what happens? This, as I said before, this bond is so strong. This binding is so strong, but once this binding is destabilized and the RNA polymerase move forward, then only the complete RNA synthesis occurs. But during the first time or the number of times the RNA polymerase falls off, the transcription factors has a strong binding affinity towards the RNA polymerase. This was the main reason of the abortive initiation. So I'll put it another way. So no, yeah, like the like the threshold is not there. Okay, it depends upon the RNA polymerase that how it will like process the transcription. So the threshold is not available yet. There might be a chance that it can go to the process of elongation without any abortive initiation. So that can also be the case, but the threshold is not available yet. Okay, any more doubts? So if not, Then we'll move on to the other two stages, that is, so first we'll see the different functions of the different transcription factors. So as we know, TF2D, the first transcription that bound to the data box, so it has a TBP binding site. Then we have TF2A, so it ensures the stability between the TBP and the TF. This is associated factors. So data box associated factors that are present in the TF2D itself. So TF2 has a stabilizing role. TF2B has affinity towards TF2F. So it is recruited before TF2F so that TF2F is recruited with the RNA polymerase. Okay. TF2F, RNA polymerase 2 recruitment. Then TF2E, it modulates the TF2H activity. Okay. Then, TF2H has a helicase activity. So it basically helps in the transition from closed binary complex to open binary complex during initiation. And then it phosphorylates the CTD and of the RNA polymerase 2. And then the phosphorylated CTD also acts as a signal for recruitment of the capping enzyme and the further RNA processing enzymes. Okay, so TF2H is the helicase enzyme, and it helps in the phosphorylation activity of TF2D. So TF2D has a phosphorylation activity that phosphorylates the carboxy terminal domain of the RNA polymerase 2. Then moving on to the elongation and the termination phase. So the two processes are nearly similar. The elongation phase is the addition of nucleotides, ribonucleotides to the growing RNA chain. Here the only difference is most of the initiation factors are shed and only the TF2F that is bound to the RNA polymerase remains, and the TF2H, because it has the helicase activity, so it will open up the front DNA chain for transcription, and TF2H, so that it will remain bound to the DNA, the RNA polymerase. Then we have some elongation factors like PTFB that stimulates protein kinase CDK9 that helps in phosphorylation of the C terminal domain. So this helps in recruitment of the capping and the splicing enzymes during transcription process only. Then we have the ELL. This basically helps in suppressing the pausing of RNA polymerase while elongation. The same thing is applied to the TF2S or S2. This stimulates the overall rate of elongation of the RNA polymerase. So these are the different elongation factors that are involved in during the elongation process. Then comes the termination process. So the complete termination process of the eukaryotic transcription is like based upon the models that are being given. So the actual model is not like understand yet that how it has been done in the eukaryotic system, but this is the model that has been well accepted now, and it is being coupled with the tailing of the mRNA. So when we have to tail the mRNA, that is the endpoint of the mRNA. So this is coupled with the tailing of the mRNA. So what happens? There are two proteins. This one is CPSF, that is cleavage and polyadenylation specificity factor, and the other is the CSTF, that is cleavage and stimulation factor. So these two proteins are recruited, and what happens, the cleavage of the RNA occurs. So for example, this is the DNA, this was the RNA polymerase, this was the growing RNA chain. So now this was the C terminal tail of the RNA polymerase 2. So here CPSF and CSTF, these two are recruited. These two bind to specific region on the RNA polymerase. So there is a site where these two protein binds, and they cleaves this RNA part that they are at cell. So they will release, this is CPSF and this is CSTF. So they release this part of the RNA, and from here the tailing enzymes are recruited, and the leftover RNA is with the RNA polymerase itself. So this is the leftover RNA. This is called as the second RNA. And here in this particular RNA, we have a 5 prime cap also. So just 1 minute. So this particular RNA that has been removed off from the RNA polymerase has a 5 prime cap. So this is not recognized by the RNAs. Okay, so this will not be degraded. But this particular RNA that is called the second RNA doesn't have a 5 prime cap. So this is recognized by the RNAs and this will be degraded. Okay, so we have the RNAs rat1 in yeast and XRN2 in humans. So what will happen as these RNAs like see the second RNA, they help in this its degradation, and now what happens? The RNA polymerase falls off from the DNA, and here the transcription is terminated. So this particular model of termination is called as the torpedo model of termination. Okay. So any doubt in elongation and termination? So in eukaryotic system, there is a case that RNA polymerase cannot exactly recognize the promoter sequences. So here the transcription factors are required so that they can recruit the RNA polymerase. Okay, so this is a feature of the eukaryotic transcription. So that is why the transcription factors are required, and RNA polymerase cannot directly bind to the DNA. Okay, and the other RNA polymerase do not have that equal abortive initiation tendencies, because the like RNA polymerase 1 is required for rRNA synthesis, so the amount that is being formed is more. Okay, and the abortive initiation tendency depend upon the strong and the weak nature of the promoter also. So how the promoter is strong and how like the promoter is weak, that also depends upon the abortive initiation. So it depends upon the RNA polymerase, the promoter type, and the number and frequency of transcriptions. Okay, any more doubts? No, there are other RNAs, like we have talked about two proteins, CPSF and CSTF, that are recruited so that they cleave off the RNA and go for RNA processing, and the other RNA that is left, that is degraded by the RNAs. RNA polymerase only have the function of polymerization. That's it. Yeah, both CPSF and CSTF bind to a polyadenylation sequence that is present on the RNA that we'll discuss in the tailing part during the RNA processing. Uh, that was the name given by the scientists that were like um, studying that. So why the name torpedo also I also don't know. I can let you know after reading it. So shall we move forward? Now this leads to the end of the transcription, eukaryotic transcription. Now we'll see about the RNA processing fall apart. So as you know, we have like why this RNA processing is needed in the eukaryotic system. So this is the nucleus, the eukaryotic cell. Here the RNA is formed. This is the RNA. So now, as you know, the protein synthesis occurs in the cytoplasm. So this RNA needs to be transported to the cytoplasm, due to which the RNAs that are present in the cell are active and they can degrade this RNA. So here we have two things. First, to make it expressive by removing the introns. We don't need the introns in the final protein sequence because the here we have the main aim of protein formation. Okay, and the final RNA transcript that leads to certain functions. And the second aim is transport without being degraded. So, in terms of expressive nature, we remove the introns by the help of splicing. The transport nature, to prevent it from degradation by the action of RNAs, we have the capping and tailing. Okay, so these are the basic reason why we need the RNA processing to be followed. So RNA processing, we have two types: modifications at the 5' end and modifications at the 3' end. So first we have the capping and cutting at the 5' end. So capping is in terms of the pre-mRNAs of the eukaryotes and eukaryotic pre-snRNAs. So these capping is linked to only RNA polymerase 2 transcripts. Okay, no other transcripts have this cap, and this capping is only due to the recruitment of enzymes due to the C-terminal domain of the RNA polymerase 2. This has been seen in various researches. Okay, so capping is not found in all the rRNAs because they are transcribed from the RNA polymerase 1, and similarly for the tRNA. It is not found in the tRNA also, transcribed from RNA polymerase 3. So it is a feature of only the RNA polymerase 2 transcripts. Then we have the cutting. This is only seen in pre-tRNA because the tRNA processing requires the cutting of the different regions to make it a functional secondary structure. Then we have the modifications at the 3' end. So it uh, gives the cleavage and polyadenylation. So this is seen in eukaryotic pre-mRNA. This is also a feature of RNA polymerase 2 transcripts only because of the recruitment of specific enzymes. And then we have cleavage and addition of trinucleotide sequence. This is ACC sequence added in the pre-tRNA. So this is at the amino acid binding site to the tRNA. This we'll see when we'll see about the pre-tRNA processing. Okay, so this is mostly done in eukaryotes, but sometimes in prokaryotes also. So first we'll start with the 5 prime and like modifications, that is capping. So capping is addition of a modified guanine base to the 5 prime end of the mRNA. Basically, this will be specific. That is called as 7-methyl guanosine. So this is the name of the cap. This is 7-methyl guanosine. Here you can see the structure of the cap. This is the guanine residue. Okay, and here is a methyl group. So this is seventh position of the guanine, and this is called as 7-methyl guanosine. This is linked to a ribose sugar. Okay, so this is the cap that has been added to this particular 5 prime end of the RNA, and it has a specific sequence, like linkage, specific linkage, that is 5 prime triphosphate linkage. So this 5 prime is linked to the 5 prime end of the nucleotide. Okay, with a triphosphate base. So how it is added, we'll see in the next slide. But this has a peculiar 5 prime triphosphate linkage that is linked to the 7-methyl guanosine cap. And the capping enzyme is like recruited to the CTD. So this is the SPF5. This is the major capping enzyme found in the eukaryotes that helps in uh, capping of the mRNA. So, what is the function of the cap? It protects the degradation of mRNA from the RNAs. It help in transport of mRNA to from nucleus to the cytosol. Why? Because certain enzymes or proteins recognize this cap, so that it will help in like moving off from the nucleus to the cytosol with the help of the nuclear pore. So certain proteins bind to this cap. Then initiation of the protein synthesis. So when during the protein synthesis, when ribosome is like bound to the mRNA, this particular cap plays a role in positioning of the ribosome with the protein synthesis starting site. Okay, so that we'll see in much in detail. So this is the particular structure of the cap that is linked with the 5 prime triphosphate linkage to the first base. Okay, so we'll see the process first. So this is the process of methylation. Okay. No, the triphosphate is not susceptible to hydrolysis like ATP, because now it has been destined to a particular function. The ATP is involved in energy hydrolysis during the like. So when this first nucleotide is added in the RNA, it is form of the trinucleotide only. Okay, it will not be hydrolyzed in like for energy consumption at all. The first nucleotide I'm talking about. The other nucleotides that are recruited in the RNA polymerization, those have two functions mainly. First is the base that is being added to the RNA chain, and the other is the energy like consumption from that particular triphosphate. No, like so. Are you asking that this particular capping is done to prevent the triphosphate to be used in energy consumption? That is your question, right? So, what happens during the procedure? This guanine base, okay, so it doesn't have a triphosphate. This triphosphate is basically from the, we'll see the process. Okay, then you'll understand how it is linked. Okay, that will help you to understand how the process is and how it is not used in energy consumption. No, it will not hydrolyze easily. So now what happens, this is the end of the 5 prime end of the RNA that have a triphosphate group. So this is the first base that have its full triphosphate groups. This is the alpha, beta, and gamma. So in my previous slides, I have also told you that if this is the sugar and the 5 prime end have three phosphate groups, this will be the alpha because this is nearest to the sugar. Then we have the beta, and then we have the furthermost is the gamma. Okay, so what happens now? There is a phosphohydrolase that releases this gamma phosphate. Okay, so this releases the pi, that is inorganic phosphate, and leaving it to only the two phosphate groups. Okay, then another GTP molecule comes that will donate its guanine residue, and the pyrophosphate is released, and this guanine phosphate monophosphate is attached to the 5 prime end of the RNA with the help of the enzyme guanylyl transferase. Okay, so this guanylyl transferase is the main enzyme because it forms the 5 prime, like the structure of the cap that has a peculiar 5 prime phosphate residue like this. So it forms a linkage between these two phosphates. So this is done by the guanylyl transferase. Now the methyl group is added with guanine at the seventh position of the guanine residue with the enzyme guanine 7 methyl transferase using the substrate edomet. So this edomet is basically the donor for the methyl group. Okay, now this is the cap. Further, this is what is called as the cap zero stage, where only the methyl group is added on the guanosine cap. Now we have the 2 prime O methyl transferase. Now this methyl group is added to the phosphate group of the first base. Okay, so now what happens? This is the second methyl group is added to the next base, that is the cap stage 1. Okay, so going back to the first step, first the phosphate group is removed, then a guanine residue is added. Okay, I'm starting it from the start again. So first, we have a triphosphate nucleotide at the 5 prime end of the RNA. So here, first the gamma phosphate is removed from the RNA chain, leaving it to only two phosphates in the first nucleotide. Then a GTP molecule donates its guanine monophosphate group using the enzyme guanylyl transferase. So this basically helps in addition of the guanosine monophosphate and linking it to the mRNA chain with the 5' 5' triphosphate linkage. Followed by this, the endomate donates its methyl group to the seventh position of the guanine residue using the enzyme guanine 7 methyl transferase. This is the major cap that is formed, and this is the stage 0 of the cap, and this is mostly present in the unicellular organisms. Then we have the 2' O methyl transferase that uses the endomate again and donates a methyl group to the first base. Okay, so this depends upon the complexity of the organism. As the complexity increases, this methylation also increases based upon the multicellularity of the organism. So now the first base is also methylated at the 2' uh, OH, so that it is not attracted to the RNAs, and this is called as cap stage 1. So this is present in multicellular organism that have low complexity. Then in the other case where the complexity is higher, much more higher, so the third base, the second base is also methylated at the 2' OH position, leaving it to the cap 2 stage. Okay, so this was the capping procedure, how a cap is installed on the 5' end of the RNA. Okay, now here, as this particular linkage is like peculiar and it is not a normal linkage that is present in the normal triphosphates, so this triphosphate, the 5' 5' triphosphate, will not be used as a energy source. This will not be hydrolyzed. So have I given the answer that you were asking for the question? So any more doubts in this particular process? So basically, there are some enzymes. Okay, so the whole RNA will not be methylated because the enzymes for recognizing different base sequences not present. That is why many proteins are there that bind to the RNA and that prevents its degradation rather than being modifying the RNA by methylation. Yeah. So they recognize basically when this RNA polymerase is like being synthesized by the RNA polymerase 2, then only certain proteins are recruited that recognize only this uh, 5' end of the RNA. So for example, this is the RNA polymerase, this is being recruited. Now here the CTD, here we have certain proteins already recruited before its elongation starts. So this is the cap binding protein. Okay, so here as soon as the cap is installed in the 5' position, this is bound to this particular protein. Further, as the RNA elongates, so here certain proteins are recruited that bind so that it is not degraded. So here the major role is played by the carboxy terminal domain of the RNA polymerase 2. Okay, so now we'll move forward to splicing. No, the capping is done when the elongation phase starts. Okay, so as soon as the RNA polymerase leaves the promoter after the abortive initiations, the capping starts. So it the all the RNA processing processes that are happening for the mRNA, those are co-translational. Yeah, sorry, co-transcriptional. So it happens during the transcription itself after the initiation process. That is when the RNA polymerase leaves the promoter sequence. So starting with the splicing reaction. So splicing is basically the removal of introns and sealing back the exons together to form a chain. Now it depends upon the intron type. Like we most common kinds are GUAG introns, and then we have GUAC introns. This depends upon the this is exon 1, this is exon 2. So this is the 5 prime splice site, this is the 3 prime splice site, and here is a branching point. So these regions are based upon the branch point that is present, and different bases present at the 5 prime and the 3 prime sites. So different type of introns have different type of structure. Okay, here we have two common types of introns, GUAG and GUAC. So most common is GUAG. So we'll be studying most probably for the GUAG only, not for GUAC. So splicing can be cis or trans. So cis means when two exons of the same transcripts are joined together. Okay. So for example, here we have exon 1, here we have exon 2. This is with intron 1. So intron 1 is removed, and this is intron 1, this is exon 1 and exon 2 linked together. This is called as cis splicing. And in transplicing, when two exons from two different transcripts are joined together, so these are called as transplicing. We'll see a bit in detail, and this is a rare process. Most common is the cis splicing. Now, this can be of two types, constitutive or alternative. Constitutive means all the exons present in the RNA transcript are basically transcribed in a mature form to the mRNA. Okay, and this is called as constitutive splicing. And in alternative splicing, what happens, some of the exons might be skipped or rearranged so that it will form a different proteins. Okay, so alternative splicing is based upon the type of organ in which the protein is being formed, because some of the proteins are bigger in some organs for some specific functions, and is smaller in the others. So this is the role of the alternative splicing, where different introns can also be retained in a particular transcript, and certain exons can also be skipped. Okay, so this was the case for the alternative splicing. Now, constitutive splicing is spliceosome mediated or non-spliceosome mediated. So, spliceosome is a specific protein and RNA structure that is being formed to remove the introns. And the non-spliceosome mediated splicing is with the use of self-splicing introns or protein mediated other than the spliceosome. Okay, so the self-splicing introns, as we have studied in the ribozyme case, where the group 1 and group 2 introns are self-splicing in nature. So they have the capability to remove out of its own from the mRNA chain. So here these group 1 and group 2 introns are basically found in the rRNA, pre-rRNA introns. Okay, and protein mediated, how the introns are removed in the RNA are found in pre-tRNA introns. So these introns are removed with the help of different proteins that are not spliceosome associated. So now moving on with the spliceosome mediated splicing. So this is the basic structure of the intron that we see. So this is the exon 1, This is the 5 prime splice site that have a GU at their like starting of the intron. Then we have a branch point adenosine. So this is an important like recognition site for different ribonucleoproteins. Then we have a sequence of polypyrimidine pack. Then we have at the end of the intron we have a AG sequence. This is the 3 prime splice site and this is the exon 2. So this is what the structure for the GUAG intron that is most commonly found. Now we'll talk about the how this particular like regions are involved in attracting the different spliceosome proteins and its extraction from the RNA. So the fundamental unit of the spliceosome assembly is known as small nuclear ribonucleoprotein. So this is called as snRNP. So here two components are there. One is the RNA component and one is the protein component. That is why it is called as ribonuclear protein. So snRNA is small nuclear RNA or the uRNA. Okay. So why uRNA? Because different components are based upon the number of U's. Okay. So U1 snRNA, U2 snRNA, U4, U5, and U6. So these are the snRNAs associated with the spliceosome complex. And the protein component, there are 20 different proteins that are associated with these RNAs and that participate in the spliceosome formation. So this is the complete process that how a spliceosome forms. So first, what happens once the protein needs to be recruited on a sequence, it needs to recognize the sequence. So what helps in recognition? First is the 5 prime splice site that was GU and here it was AG if I'm not wrong. Yes. So the 5 prime splice site is recognized by the U1 snRNP. Okay. The 3 prime splice site is recognized by the U2 AF, that is accessories factor. So U2 AF recognizes the 3 prime splice site. Then the branch point, that is the adenosine, that is most conserved sequence, is recognized by the branch point binding protein, that is BBP. Okay. So when these three proteins, ribonucleoprotein, bind to the particular regions on the mRNA, this is called as the commitment complex. So whenever these three proteins are bound to the mRNA, that means the now the mRNA needs to go to the splicing reaction and it cannot go away from that particular process. Now this BBP recruits a U2 snRNP. So this U2 accessory factor is different from the U2 snRNP. Okay. So now U2 snRNP is recruited and that binds to the branch point. So the BBP is released now. So now this U2 snRNP has high affinity towards U1 snRNP. So they have a close affinity towards each other. So they basically like gets attracted to each other. This is called as the pre-spliceosome complex. Okay. Now there is another recruitment that is U456 snRNP. So these are two snRNPs, U4 and 6 are together and the U5 is separate. These three are recruited together and with the release of U2AF. Okay. Now what happens? These three snRNPs that were recruited, they basically combine with U1 and U2 snRNP and forms a branch between the 5 prime splice site and the branch point. So now the U1 snRNP released, this is called as the spliceosome complex when all the snRNPs are formed in the mRNA intron. And then the U1 snRNP is released and U5 and U6 are bound to the 5 prime splice site. Now what happens? U4 is released from this particular structure, so that now the U6 can act upon the 5 prime splice site and it can cleave off this particular site. So here this U6 and U4 complex cleaves the 5 prime site and U4 is released from the RNA. Further, this particular structure forms a closed loop of the RNA called as the lariat structure. And this was the first two consecutive transesterification reactions that we have studied previously also. This lariat structure was formed. Now comes the this was the C complex because the 3 prime splice site is still attached. Now this U6 U5 complex is a endonuclease. So this cleaves the 3 prime splice site, leading to release of this particular lariat structure bound to the uh, U2, U5 and U6 SNRNPs and the combined chain of the exons. So once this U6 and U5 cleaves the 3 prime splice site, this end of the 5 prime end of the exon, 3 prime end of the exon has a nucleophile attached to it, to the phosphate group. So this nucleophile adds a nucleophilic attack on the 5 prime end of the this particular base present at the 5 prime end of the this uh, second exon, leading to formation of a complete chain of the RNA binding the exon 1 to the exon 2. Okay, leaving the intron bound to the snRNP behind. So this leads to extraction of the intron from the uh, RNA chain. This was the spliceosome machinery and how different snRNPs helps in the removal of the intron. Now we have a second point, that is transsplicing. So here the splicing machinery for the constitutive splicing ends, and the self splicing introns we have studied previously also. So you can go back and check the lecture there. And then moving on to the transsplicing. So anybody has any doubt till here? So basically, the basic advantage of this particular spliceosome complex is specificity. Okay, because the self-splicing introns are directly basic function as RNAs, so they can degrade the different RNA present in the system. So that is why, with the increase in complexity of the organisms, the levels of the group 1 and group 2 introns also went down. Okay, so that is why the specificity leading to splicing increased and the spliceosome complex formation increased. This is the main advantage of the spliceosome complex. Yeah, the basic advantage is this only. Hmm. So all the exons are removed, and like this was the basic structure, that how like the splicing mechanism follows. Okay, now in case of Alternative splicing also plays hand in hand. Only the term are different, but the process remains the same. Okay, so these are already like in process. So the introns are removed continuously. So moving on to the transplicing. So the So moving on to transplicing. So this process is found in only the lower eukaryotes like trypanosoma. And here, as we discussed earlier also, two different transcripts have exons and they are linked together. That is why it is called as transplicing. And the intron that is formed is a Y-shaped branch structure rather than a lariat structure, and this is mediated by the splice leader RNA. So what happens as this there is a pre-mRNA. So there are two pre-mRNAs. This is the splice leader mRNA. Here we have a 5 prime splice site. Okay, and the 3 prime splice site is present on the different mRNA. Now this splice site provide the exon 1, which needs to be joined to the exon 2. Now, first esterification reaction occurs where this branch point will be present on the second transcript. Now, this branch point has a 2 prime OH. Okay, that have a double like a lone pair that leads to nucleophilic attack on the this particular splice site. Now, this 2 prime OH attacks on this particular 5 prime splice site and leads to its excision from the exon 1. So now exon 1 have a 3 prime OH free. This 3 prime OH leads to a second transesterification reaction and a nucleophilic attack on the 3 prime splice site. So this leads to the excision of the like joining of the two exon chains and leaving a Y-shaped intron behind. Okay, so there are various proteins involved during the transplicing process, but that is like too much deep information for this class. So this was the like full process, how the basic process happens for transplicing. Now moving on to the alternative splicing process. So, as we discussed, like in different organs, different proteins are formed in like different amounts and different structures. So, like this particular alternative splicing process helps in forming different length of proteins of the same protein in different organs. So, this is a particular like process that have been shown in figure A, where the 5 prime splice site, this is the 3 prime splice site of two of a intron that needs to be retained, and this is the poly A site. So this is the primary transcript with the cap and all the sites. Then we have the cleavage and generally the splicing is not being observed in prokaryotic organisms. Okay, because it's a energy consuming process and prokaryotic organism cannot like spend too much energy on these processes. So they have a very much simpler system. That is why the operon concept came, that different genes that are involved in a similar process are linked to only one promoter. So they do not have to recruit the RNA polymerase for each and every promoter, and only a single protein single transcript is formed for different proteins. So this can be the case where the polycystronic mRNA do not follow the alternative splicing wala path. Okay, So here, after the cleavage and polyadenylation, we have two types of splicing. Here, the this particular splice site is not used, and the poly A signal is like added to this. And here, this particular splice site, this particular region is spliced, and the rest is followed. So what we have is two types of transcript that have been followed different splice site, so that the different length of the mature mRNA is formed, leading to different protein sizes. Okay, so there are different ways where the alternative splicing can work. So first is the alternative exon. For example, this dark green one is being removed from one of the transcript, but it is present on the full mature transcript. Okay, then we have alternative 5' splice sites, such that this particular exon, the dark green one, can be skipped, and in one exon only there is two 5' splice site, so that this exon can be split into two and linked to form two different transcripts. Then we have 3' splice site, some similarly to 5' splice site. In one exon, we have two 3' splice site, leading to the distribution of one exon in either of the two transcripts. Then sometimes the intron retention is also there, leading to more complexity of the protein involved in a particular process. So in one of the transcript, the intron will also be retained, and in other, no intron will be retained. Then we have mutually exclusive alternative exons. So in one of the transcript, only one exon will be there, but not the second one. But in the other, the second one will be there, but not the first one. So this leads to different functions of single protein in different organs or different tissues. Okay. Then we have alternative promoter and the first exon. So the this leads to the formation of different transcripts, and based upon the function, it will be like different in different tissues. Then we have alternative polyasides on the terminal exon. So this will lead to formation of a single transcript that have a shorter size with the poly A tail, and this leads to a shorter protein. And similarly, this will have a longer size with the longer poly A tail. So these are some of the ways with which the alternative splicing works. Now we will take an example of the alternative splicing with the calcitonin gene transcription. Okay, so calcitonin is a like hormone that is released from the parathyroid gland and help in calcium metabolism and calcium homeostasis inside the body. So this is the primary transcript of the calcitonin molecule. We have the green ones are the introns, and the yellow ones are the introns, and green ones are the exons. And this fourth exon, is the major calcitonin uh, catalytic site, and we have here two poly A sites. The last way which we discussed, so these are the two poly A sites that we have, and that leads to different transcripts. So in thyroid gland, we have the first poly A site here, leading to the smaller calcitonin molecule, and the first full length transcript leads to the formation of a longer transcript, and leading to formation of the CGRP, that is in the brain. So this is a protein that calcitonin gene related peptide that helps in production of the like calcitonin to uh, during the production process or during homeostasis. So this is the case where the same molecule which is alternatively spliced leads to two proteins helping in different processes. Okay, so we'll see more example of alternative splicing in the next class, but this was the first example where calcitonin primary transcript leads to the formation of calcitonin and CGRP in brain. So Do you have any doubts till now? So like this depends upon the specific proteins present in that particular organ. Okay, so there are specific proteins for the adenylation, sorry, for splicing out that particular exon only. So these are tissue specific as well as protein specific proteins that are present in particular organ. No, the like the different the spliceosome complex is only help in recruiting that particular region. Okay, sorry, removing the intron. But what happens when a specific organ needs to retain a particular transcript? For example, in thyroid, this thing happens. So how this thing happens? There are certain transcriptional modifications. Okay, that leads to retention of this particular thing. And so what happens here after this? The spliceosome will not be able to recognize any of the introns. So that will help in retaining that particular intron or the polyA site. What will happen? This site will get activated, and here the cleavage will be there in thyroid. But in brain, this site, this that is the polyA site, the first, this will be masked by the post-transcriptional modifications by different proteins. So what will happen now here? The spliceosome cannot recognize this, and sorry, the polyate proteins cannot recognize this, and this will not lead to the cleavage of the mRNA here. Now the mRNA will be cleaved here. Okay, so these things are linked to the post-transcriptional modifications. Huh? Yes. So this leads to the end of today lecture. We'll meet you tomorrow from 3 to 5, and if you have any questions you can ask me there. Okay. So we'll switch off the class now.